Hello, good afternoon, everyone. We have a lot of things to go through today, so let's get right into it. Previously, we were talking about macromolecules and how they are composed of repeating building blocks that you can join together through chemical reactions, and you can make a long chain of polymers for each of the macromolecules. And today we are gonna hopefully finish the we talked about carbohydrates as a macromolecule last time. So today hopefully we'll be able to finish the other three, which includes lipids, proteins, and nucleic acid. Not a lot of notes today, but let's start with a quick one here to help us summarize the different types of lipids. Okay, so please take out a piece of paper and we're going to put lipids right in the middle. So there are different kinds of lipids, 4 types that we're going to look at specifically. And the lipids are a little bit different from the other three macromolecules, because instead of a long train of like repeating units, you don't have to copy this next part. Like for carbohydrates, take cellulose, for example. It's like glucose connected to glucose connected to glucose, and then it repeats like hundreds and hundreds of times, right? But and this is the same theme for things like proteins and nucleic acid, but that's not the case for lipids. Okay? Rather than chains of repeating units, lipids are made up of specific components, and different lipids have different combinations of these components. So let's start with triglyceride. Triglyceride is fat. Okay, it's just a fancy word for fat. And the function of fat is for long-term energy storage. Okay. And we also need to use fat for insulation and protection of organs. So to insulate an organ means you are gonna keep it warm, and you can protect them by kind of providing some cushioning or some like bubble wraps around the organ. So if you fall down, you're not gonna hurt your kidney or something like that. And that's those are the key main function of fat. Okay. Now, how do you make a fat molecule? Well, fat needs 2 components. We have this thing called the glycerol, and the glycerol is connected to 3 fatty acids. And I'll tell you what fatty acids are later, okay? But you need 3 of them. And that's why the name is triglyceride. Tri means 3. Like triangle has 3 sides, tricycle has 3 wheels. Triglyceride has 3 fatty acids. Okay, so that's pretty much all you have to know for fat at this moment. This is just an overview. So when we go through the PowerPoint slides, it will like make more sense to you. Another lipid that we're going to be looking at is phospholipid. Phospholipid. Phospholipid is the major component of cell membrane. We know what a cell is. It's the basic unit of life. And the cell looks like this, and the outer edge of a cell, if you would, that would be the cell membrane. and that is made up of phospholipids. Phospholipids. How do you build a phospholipid? You need glycerol as well. And 2 fatty acids. 2 fatty acids. And a new thing here called the phosphate head. Phosphate head. All right. Give you a moment to catch up. Any questions? All right. Does anybody have another example of a lipid that they can think of? Something that you probably heard of. No? That's okay. Another example of lipids would be wax. So wax are very good for forms water repelling surfaces. So the skin of an apple, right? Your skin, your hair, they are not gonna let water go through, right? So there's a bit of wax there. And to make wax, you need this fatty acid again. And on the fatty acid, there are a bunch of alcohols on it. Okay. So OH, when a molecule has OH, it actually is referred to as an alcohol. Alcohol is not necessarily like drinking alcohol, right? The drinking alcohol is ethanol, but there are other kinds of alcohol. Like rubbing alcohol is propanol. And then there are things like, methanol, right? Your windshield fluid is a type of alcohol, right? Called estradiol, right? So all these things are different types of alcohol. And chemically speaking, an alcohol has OH on it, an oxygen connected to a hydrogen. Is it recommended to have a reference book? Can you please tell me the name of the book and edition? Yeah, you don't need a reference book really, right? Like most of the stuff here are all contained in the PowerPoint slides. But if you do want to read about it, I have this in one of my earlier videos, but if you go to eCentennial, each topic has a link to an online textbook. Okay, like I mean, if you need a physical textbook, just go to the library, like school library or like your local library. Any introductory biology textbook would have these things that I'm teaching you. Okay, but if you want the online textbook, you go to the lecture. Let's say lecture 2 here. Study resources, and you can watch all these steps on the recordings, okay, if you are not able to follow. It says supplementary reading, right? So you click on those, it will take you to an online textbook. All right, Sheila? Okay, besides wax, we also
Thank you
have,
so much.
yeah, no problem. Besides wax, we also have steroids. Steroids, okay? A lot of people, when they hear steroids, they think of, steroids that athlete use to build muscle, but we actually have steroids in the body, okay? So the function of a steroid is, so for example, we have cholesterol. cholesterol is a steroid, and cholesterol is used to make part of the cell membrane as well. Most of the cell membrane is made up of phospholipid, this guy, but there is a bit of cholesterol in there as well. Also, sex hormones is an example of steroid. Does anybody know the name of male, female sex hormone? Start with the male one. What's the male sex hormone?
Testosterone.
Testosterone, that's right. That's a steroid. Okay, if it ends with like ONE, it's some kind of steroids usually. What about the female one?
Estrogen.
Yep, that's one of them. And there's one that starts with P.
Progesterone.
You got it, progesterone. Okay, so these are steroid hormones. Not all hormones are steroids, okay? Some hormones are protein hormones, as you will learn them later on when we get to the protein section. Sarah has a question. What does it mean to have high cholesterol? Yeah, so cholesterol, again, there's this bad association people make with cholesterol, right? And it's true, right? There are good cholesterol and bad cholesterol, right? So when a person is high in cholesterol, they're talking about there's a high content of like usually bad cholesterol in their blood. All right, so you can look it up if you're interested. It's called HDL, that's the good one, and then LDL, that's the bad one. All right, you'll learn more about cholesterol and its role in cardiovascular diseases next semester. Okay, but for the most part, we need cholesterol. Right? We need some cholesterol to mix the cell membrane, and we need it to make hormones such as these ones that are listed here. Okay, so that's an overview of lipids. All right, keep this picture next to you as we go through the PowerPoint slides, and you can kind of. Oh, I totally forgot. One more thing here. The steroids. We have to look at how to build one, right? So the defining feature of a steroid is that it has carbon rings in it. Okay, it has 4 carbon rings to be specific. Okay, and the rings are it could be hexagon, it could be pentagon, it doesn't matter, but there are 4 carbon rings. And in addition to the carbon rings, we have some other groups that are attached to it. Okay, so these other groups, they are called functional groups. You will learn more about functional groups in organic chemistry next semester. But basically, functional groups, they are basically group of atoms that give certain properties to the molecule. So, for example, OH makes it more soluble in water. Okay, alcohol soluble in water. This makes it polar, and so on and so forth. You don't have to memorize it, okay? Like when you learn it in organic chemistry, you will know the properties. But like that's what a steroid is. 4 carbon rings, and then with these things that are added onto it, and give it special properties. That's basically what you have to know. All right. So here is our PowerPoint slides. We are gonna go through lipids. Chemistry of lipids. Lipids contains C, H, and O. For all the macromolecules that we're going to be talking about, the carbohydrates, the lipids, the proteins, and the nucleic acid, all of them will have C, H, and O in it. Okay? Carbon, hydrogen, oxygen. However, if you have the piece of note that we just talked next to you, you can see that some lipid contains trace of P, phosphorus. Which one contains phosphorus? Can you see in the note? I'm just gonna go get my pencil case here. Which Yeah, go ahead, Elvis.
Phospholipid.
That's right. The name tells you, right? The name has the word phospho in it, so it contains phosphorus. Unlike other macromolecules, lipids are built with components. That's why we drew that thing for you, right? You know, we need a red rectangle, we need 3 of the blue rectangles and whatnot, right? So each of those components is what we need to create the lipids. It's not like the other 3 macromolecules where it's the same thing repeated over and over and over again. Right? So that's a key distinction you have to make. And lastly, all lipids are hydrophobic, which means they cannot dissolve in water. What is the opposite of hydrophobic? That's right, Kyle, hydrophilic, right? And Sarah as well. Very good. So let's take a look at the different types of fat. Okay, fat is an example of lipids, right? 4 different types of lipids. Okay, don't confuse lipids with fat. Fat is a type of lipids. There are other types of lipids as you know, as you have on the sheet that you took down the note. So triglyceride, it's made up of 1 glycerol. That's this guy right here. That was the red rectangle, so to speak. And then you have the 3 fatty acids. Now, how do we connect them together? If you have been reviewing the notes, you should remember to build macromolecules, we need to remove water to connect things. We need to remove water molecules to create a covalent bond to connect them together. The process is called dehydration synthesis, and we learned that last week. So here on the glycerol molecule, right, we have an OH, OH, and OH. Some people call this the ho-ho-ho molecule, makes it easier to remember. And on the fatty acid, there is OH at the end as well. So in order for us to connect these together, your body will remove the H and the OH and create a bond, a connection. So there is a bond, there's a bond, there's a bond. Because there are 3 fatty acids that needs to attach onto the glycerol. For every fat molecule that you build, you will release 3 water molecules. Okay. Of course, you can break the fat back down into glycerol and fatty acid. And that's what you do when you need to burn the fat for energy. And the opposite of dehydration reaction would be hydrolysis. Okay, we took a note on this last time. Okay, hydrolysis is to break the bond by addition of water. Food source. There's no shortage of fat in most diet. Of course, there are healthy kind of fats, and then there are less healthy kind of fat. Generally speaking, if it's like fast food kind of fat, it's not really good. But you know, fish, avocado, nuts, they generally have good fat content in them. Again, like I said, there's no shortage of fat usually in people's diet. Now, talking about healthier fats and less healthier fat, then we should look at saturated versus unsaturated fat. Which one do you think is healthier? Let me do a poll and see what you think, okay? Do you think A, saturated fats are healthier, or B, unsaturated fats are healthier? What do you think? Okay, very nice. Of the people who've answered so far, we have an even split again, just like most of the things I asked you guys, right? It's always 50-50. Well, let's find out which one is not as good, okay? So for saturated fatty acid, it's made up of 1 glycerol, which is like the red rectangle, right? Like the ho-ho-ho molecule. And you are gonna have 3 fatty acids, okay? The fatty acids. So what exactly is a fatty acid? Well, if you go back to, I mean, you don't have to go back, you just look at the screen. These are all fatty acids, okay? And the fatty acid besides like this thing at the end, you basically have C and H and C and H that are keep on repeating, right? So, this one has 4 carbons, but usually, the common fatty acids that we have in the body, they are much longer, okay? 10 to 16 carbons are possible. So carbon is able to form 4 connections with other atoms. And in the case of saturated fatty acid, the carbon is maxed out with its connection. So there's 1 connection here, 2, 3, 4. There are no more rooms to make additional connection. In other words, it is bound to a maximum number of hydrogen atoms. No more rooms. Right? That's what makes it a saturated fatty acid. It's saturated with hydrogen, basically. So when the fatty acid is saturated with carbon, the molecule, the tails, they go zigzag up and down, but for the most part, it's straight. So if I draw a glycerol molecule here with 3 saturated fatty acids, and then I can have another one right next to it. Okay? Because the tails are straight, it's very easy to put them close to each other. Okay? It's like, asking people to line up with their arms down. They could stand shoulder to shoulder, very close together, right? They get packed close together. And that's kind of the problem with saturated fat. They pack quite nicely together, so they're solid at room temperature, and they would pack equally nicely in our body. So if you have a lot of saturated fat in your diet, they tend to build up in the inside of arteries, causing them to be hardened and perhaps clogged later on. Okay, so we don't want to have too much saturated fat in our diet. Most animal-derived fats are saturated. Okay, derived means it came from animals. Okay, so butter is an example of saturated fatty acid. Things like lard, that's saturated fatty acid, and people use it to make pastry, right? Make it more fluffy and what's the word I'm looking for? Like a flaky, right? But it's saturated fatty acid. It's not that healthy. On the other hand, we have unsaturated fatty acids. So to make up unsaturated fatty acid, the components that you need include, once again, the glycerol molecule, and then you would have 3 fatty acids as well. So there's 1 fatty acid, 2nd fatty acid, but at least one of these fatty acid has to be unsaturated. Okay, so in this case, I only draw 1 unsaturated fatty acid. Unsaturated fatty acid is different from saturated fatty acid because it has a band in the structure. Okay, so it's in order for the whole thing to be an unsaturated fat, one of the fatty acids needs to be unsaturated. Okay, so people sometimes confuse. They get confused between saturated fatty acid versus saturated fat. Okay, the fat is the whole molecule, glycerol plus the fatty acids. If one of the fatty acids is unsaturated, then the whole fat molecule is considered unsaturated. So you can have a molecule that looks like this, that's unsaturated. You can have another one, okay, glycerol, and then you have a saturated fatty acid, unsaturated fatty acid, and then another unsaturated fatty acid. And this whole thing would still be unsaturated. Okay, so if at least one of the blue things are unsaturated, then the whole thing is unsaturated. Okay, you can have 3 unsaturated fatty acids, then it would make it like a polyunsaturated fat. Okay, so what exactly does it mean? Well, in this case, over here, this last fatty acid is unsaturated. There is what we call a double bond. Double bond. So I told you carbon can bind to 4 things in total. So if you look at this carbon right here, it's bound to 1 thing, 2 thing, and a 3rd thing. So that means it actually has space to make one more connection. Right? We can fit more hydrogen onto it, right? And that's what an unsaturated fatty acid is. Now that the presence of that double bond, of that, equal sign, if you would, creates that kink, that bend in its structure. So now if you try to pack these things together, it's not as easily, cannot be done easily. Okay, so like this is one fat molecule, okay, and then it has the thing sticking out. So if I need to put another one next to it, right, this is going to be in the way. You can't get really close to each other, right? So back to the lining up analogy, if you stick your arm out, okay, like then this guy is not going to be able to get as close to you, right? And like let's say this guy stick the arm out again, right? So now it's not packed compactly, and so it tends to be liquid at room temperature. So most plant-derived fats are unsaturated, and generally people cook with unsaturated fats, right? It's a little bit healthier. What kind of cooking oil do you guys use? Let me know. Anybody cook here? What kind of oil do you add to your pan? Yes, olive oil. That's an example of unsaturated fat. Avocado oil, exactly, right? Canola. There's a bunch of these ones, right? Now, some people use coconut oil. Okay, coconut oil, if you ever use it or if you ever seen it, it's like it's not completely liquid. It's more almost like a white, foamy kind of paste. Okay, so even though coconut is plant-based, but it actually has a very high saturated um, fats content. Okay, so it goes back to the theme that I was trying to tell you at the beginning of the course. Even though we try to fit things into different categories, but sometimes there are exceptions. Okay, and coconut oil is an exception here. Okay, so saturated fat, not too good. Unsaturated fatty acid, it's okay. So there are also essential fatty acids. Essential means we need it in the body, but we cannot make it ourselves. Our body cannot create them. So the only way for you to obtain them is to eat them. An example of essential fatty acid is something called omega-3. You probably have heard of that, okay? Besides omega-3, you probably know other numbers, right? Omega-3 and then omega, what's the other ones that you heard of before?
6,
Yeah,
9.
exactly, right? 3, 6, 9, right? It's like reciting the multiplication table, right? So 3, 6, 9, these are the good ones. And examples of it would be like EPA and DHA. You don't have to memorize these names. You can get them from, eating fish, like avocado, and certain nuts are pretty high in these. Some eggs are advertised to have extra omega-369, right? That's what they look like. The 3 refers to the location of the first double bond. So if carbon number 3 is the first time you see the double bond, then it's an omega-3. If the carbon number 6 is the first time you see the double bond, then it's an omega-6. Pregnant women are usually advised to take omega-3, 6, 9 supplements. They are essential, and it helps with the fetal brain development. Okay, people with cardiovascular diseases are also sometimes advised to take these fatty acids because they are good for the health of the heart. But generally speaking, don't start taking supplements unless your healthcare provider advises you to do so. There are a lot of false claims out there, right? With some of these supplements, and you don't want to be taking the thing. Number one, when you don't need it, then that's just a waste of money. And number two, like it might actually harm you if you take certain things that is not suitable for you, right? So always consult your doctor. So here is just a side-by-side comparison of saturated and unsaturated. Can you tell which one is which? Like which one is the unsaturated, right? Is it A or is it B? Which one is unsaturated? Tell me. Here we go. I'm looking for the unsaturated. Okay, very good. The answers are pouring in, and everybody who chose something said that this is unsaturated. Okay, this guy is unsaturated, and you'll be absolutely correct. You're looking for the equal sign, right? I keep on calling it equal sign, but it's actually a double bond, right? That's the telltale sign that it is an unsaturated fatty acid. And you can see there are all these kinks, all these bands in the tail. That's also another sign that it's an unsaturated fatty acid. Now, trans fat is like the worst kind of fat, okay? And how you get trans fat is if you take unsaturated fat and you add hydrogen to it, then you get saturated fat. And that's how you make margarine and shortening. Like, I get it, like avocado oil or what you guys say, like olive oil, they are healthy, but like you can't really spread them on your toast, right? Okay, like I mean, I get it. Some people like dip their bread in the avocado oil or olive oil or whatever, but like that's not what most people want to do, right? So they as in like the food industry found a way to take vegetable oil, which is liquid, and convert it into a spreadable form. And this process is called hydrogenation. And the byproduct of hydrogenation is trans fat. Okay, now before we get to that, these kind of hydrogenated fat are highly processed and they're not too good for you. Do you guys know that margarine product is just like I can't believe it's not butter? Right? You probably know what I'm talking about, right? It's true. It's not butter, because butter is like the natural form of saturated fat. This one is like converted, right? It's processed to be in that spreadable form. And in this process of converting it, you generate trans fat, which we cannot metabolize, we cannot break down because we don't have the enzyme to do it. Okay. Every reaction that happens in the body requires an enzyme, and more on that later. But we don't have it. We can't break down the trans fat. They just build up in our body continuously. Okay, it stays there for a very long time, and it's linked to increased risk of heart disease and diabetes. So trans fat are like the worst. But trans fat are widely used in processed food because they increase the shelf life. Many fast food chains, they use trans fat for their food because it makes them taste better. And that's why these food, if you consume a lot of it, is gonna increase your risk of getting heart diseases and diabetes in long term. So because the trans fat is so bad that the Canadian government actually banned the use of artificial trans fat in 2018. Okay, so like if it's like occurring naturally, then it's okay. But if you are adding it on purpose to the food, then that's not allowed anymore. Okay, so it's a little bit healthier nowadays to eat your fast food than it used to be. There is a question: does adding hydrogen turn it into? Okay, yeah. So adding hydrogen turns it into saturated fat, but um, there is a byproduct in the process, and the byproduct is trans fat. Okay, like you're not intended to make the trans fat, but it just happens to be there as part of the process. Does it answer your question? Great. That's it for fat. Another type of lipids, if you have the sheet right next to you, again, it's good to, take a look side by side in comparison to help you remember these things. This second one is called the phospholipid, and this is the guy that has the phosphorus in it. Everything else is just CH and O, but this guy has the phosphorus in it. Now, to build the phospholipid, we need the glycerol, which you will see as a red rectangle in the notes I gave you. There is going to be a phosphate head, okay? Phosphate head. Now, you haven't, like my other classes told me, you haven't got there yet in your chemistry, but you will soon. Phosphate is PO4 -3. Okay, so it's hydrophilic because of the negative charge. Okay. On the other hand, you have two fatty acid tail, and these are gonna be hydrophobic. So basically, you can draw the phospholipid like this. Usually that's how people draw it. One of them is saturated, the other is unsaturated. It's also possible that both of the tails are saturated. It doesn't really matter. The point is that it has a hydrophilic head. Hydrophilic head, and it comes with two hydrophobic tails. And that's what makes it a special molecule. Okay? The head is able to bind to the water, and the tail is able to bind to other lipids. Actually, you are familiar with molecules like this in everyday life. Okay, I mean, like you probably don't think about it, but you are using it when you are washing your dishes. Okay, when you have a greasy dish and you try to wash the grease off with just water, it's not going to clean the plate. Right? And it makes sense because grease and water don't mix together. One is hydrophilic, one is hydrophobic, right? But when you add detergent, the detergent actually has the same structure as phospholipids. It has a hydrophilic portion and a hydrophobic portion. The hydrophilic portion will bind to the water, and the hydrophobic portion will bind to the grease, and it allows two things that normally don't mix together to bind together, and you can wash it off the plate. Okay, so that's what we use it in everyday lives. However, phospholipids are a major component of the cell membrane. We are gonna go into a lot more details about phospholipids and cell membrane in lecture 4, but for now, you can think of the cell membrane as phospholipid bilayer. There are two layers of these things, basically. You can visualize like a peanut butter sandwich. The bread on the outside and the inside, those would be like the hydrophilic heads, and then the peanut butter in the middle, that would be like the tails. Okay, sandwich in between, and that's all you have to know for now. Okay, again, much more details to come later on. Next, we have wax, which covers exposed surfaces of plants. They protect and lubricate skin and hair. We know that already. Also found in honeycombs. A lot of cosmetics are, derived from honeycombs. Bee wax. There are bee wax, chapstick, for example, lip balm, and all sorts of stuff. It's like they make it more organic, right? Supposedly. And it consists of long fatty acids connected to alcohol. Exactly what we wrote down in the summary. Okay, that's all for wax. And steroids. Right? How do you know something is a steroid? Well, a steroid has these carbon rings in it. This particular steroid is cholesterol. Right? And there are different functional groups attached onto it. There is an OH here. There are all these things that you don't have to know what it means at the moment, but they're like accessory. They're like add-ons to the carbon rings. If you add different things, it's going to behave differently. Okay. We need cholesterol because it's part of the cell membrane. Much of it is the peanut butter sandwich, right? Much of it is the phospholipids, but every now and then you're gonna have cholesterol. Okay, back to the peanut butter sandwich analogy. Like every now and then you would find a peanut in the peanut butter jam, right? Okay, it's not all over the place. There's a bit of it. That's basically what cholesterol is. Cholesterols are also needed to make steroid hormones such as estrogen and testosterone. You can see they're very similar in structure. Okay, like, there's the OH, there's the OH, there's CH3, CH3, and you have the carbon rings, and then there's like some subtle differences at one end. Okay, so if you see, if I show you a picture with 4 rings in it, and I ask you what it is, you can safely say that it is a steroid. Okay, you wouldn't be able to tell whether it's testosterone, estrogen, or cholesterol. I don't expect you to memorize its structure, but I do expect you to know when you see 4 rings, and the safest bet is that it belongs to a steroid. Okay, so cholesterol is an example of steroid. All right, we are on a tight schedule today. I'm really sorry because there's a lot to go through. And since you guys missed the Mondays, right, we do have to speed things along. So I'm going to skip these concept track questions. I'll post the answer for you later on, okay? You can do them later on. Let's move on to proteins. Okay. Proteins. Mainly composed of, again, the triplet, which is C, H, and O. In addition to CH and O, they also have nitrogen. Nitrogen and trace of sulfur. S is sulfur. Okay. Trace means a little bit, not a lot, just a bit, just like trace of phosphorus for lipids, right? Now, the monomer of protein, the building blocks of proteins, they are amino acids. Okay, amino acids. Now, I do want to go here and just quickly write this down for you because I know it's like a lot of things here and there and people are getting confused perhaps. So I want to do like carbohydrate, lipids, proteins, and nucleic acid in a bit. All right, so let's write down the elements and monomers here. Now you need to expand this chart when you're studying, okay? Add like a column for like food source, add a column for like, biological function and so on and so forth. But we don't have time to summarize it all today, but I'll do 2 columns for you. Okay, quickly, anyone. For the elements of carbohydrates, what are the different elements that makes up a carbohydrate? Do you guys remember?
Yeah, cellulose.
Oh no, cellulose is an example of carbohydrate, but like what are the elements that build it up?
Oxygen.
Uh-huh. Yeah.
Carbon.
Yeah. And hydrogen, right? Yeah, a lot of people are writing it in the chat as well. Okay, so CH and O, very good. What about lipids, guys? What are the elements of lipids?
C also.
I think it's the same thing.
Yeah,
It's the same thing. That's right. Plus a little bit of P. Very good. Not phosphate, but phosphorus. Okay, phosphorus. Phosphorus is just P. Phosphate is PO4. Okay? Again, your chem teacher will explain the difference, but this is trace. Okay, trace of it. Okay, I'm glad we are making this table, okay? Because I don't know, like a lot of you probably could use a summary. What about for proteins? Okay, I literally just told you. So help me out again. What are the elements here?
Carbon, hydrogen, oxygen, and nitrogen, and a little bit of sulfur.
A little bit of sulfur, that's right. Okay, so it's always CHO, okay? Just remember like cho, and then for lipid, there's a bit of P, and for protein, it is nitrogen and a little bit of S, sulfur. We good?
Yes.
Thank you. Monomers. Okay. Again, like I don't expect you to memorize all these things on the spot, which is why I have the study guide for you. And did I already tell you about writing the quiz? Have I tell you about that yet or no? I don't think so. Maybe not. Okay.
No.
So maybe I'll take a moment to talk about this. Your first quiz is going to be next Monday. Okay. We have class on Monday, so you're not doing the quiz during class. The quiz will be available to you from 6 a.m. all the way to 11:59 p.m. I will post more information about the quiz later on today, like how many questions are on it and that sort of stuff. Okay. But I just want to tell you this. I encourage you to create a help sheet for yourself, a cheat sheet, whatever you want to call it, okay? 2 pages, front and back, okay? There are some things that are particularly difficult for you to memorize, for example, put it on that sheet, right? So you can use it during the test. It's not cheating, okay? At least I'm just talking about for my course, okay? I don't know what the expectations are for your other courses, but for my course, it's okay that you use a sheet to help you, okay? Now, you will be using the lockdown browser, so you won't have access to the internet, at least not through the device you're doing the quiz on, okay? And if you have like digital notes on the computer, for example, and you're using the same computer for the quiz, you can access them, okay? So you gotta write the cheat sheet out, okay, and have it next to you. If you print out your notes, you might have your whole binder of notes. I don't recommend that, okay? I don't recommend you just trying to find the stuff in all the notes because you won't have time, okay? There are a lot of questions on the quiz, and if you're searching things up as you go, you will run out of time very quickly, okay? But if you create that help sheet, you know exactly what's on it, right? You still have to study, you still have to memorize things. But for some things that are particularly difficult for you to remember, you can have it written down on a piece of paper. Does it make sense? Okay. So do take advantage of that. The questions are designed in a way that that has taken that into consideration. Okay. Like I'm not like naive, right? I know if I tell you like don't use your notes, like some people are still gonna use it, right? So I'm just gonna flat out and say it. Yeah, by all means, use your notes, okay? But use your notes wisely. Use it in a smart way. Okay. You can't flip through the notes and find everything. You just won't have time. Anybody have any questions regarding that? All right, now again, that's only for my course, okay? Don't go and tell your art teacher, oh, my biology teacher said we can do this for your class. I didn't say that, okay? Just for biology. Back to this monomer. What kind of monomer do we have for carbohydrates?
Glucose.
Yes, glucose, and very good, Sophia, add to your answer. Glucose, fructose, galactose, right? And generally, we call them monosaccharide. Okay, monosaccharides. Remember, I said last class, you know, they are single and they are ready to mingle, right? They can come together and form disaccharides, and ultimately, they can form the polysaccharide. Okay, we do not have monomers for lipids. Not applicable. Na. Okay, do not have it because lipids is different from the other kinds. Lipid has components, right? Okay, a monomer is something that you keep on using repeatedly over and over and over again. That's not the case for lipids. Okay? So for proteins, which is the fresh one that we learned, it is called amino acid. Amino acid. In carbohydrate, there are 3 kinds of monosaccharides. For amino acids, we have 20 types. 20 different amino acids. Don't worry, you won't have to memorize all of them. Okay. In fact, you don't have to memorize any of them. So that's the building block for proteins. Okay, so I will post this. So even if you don't have time to copy everything, you can catch up afterwards. Of the 20 amino acids, 9 of them are essential. Do you remember what essential means? We'll talk about essential in the context of lipids. These are things that we cannot
produce.
That's right, we cannot produce, but we need them. Okay? We need them to survive. So here are 9 essential amino acids. okay? You don't have to memorize them. I'm just showing you. So these things we absolutely need in the body, but we can't really produce them. If you have a relatively balanced diet, then you got your amino acids covered. Okay? They are found in pretty much basic things that we eat. Okay, it's nothing obscure. If you eat meat, you're covered. Okay? Even if you're vegan and don't eat meat, as long as you have a good source of plant-based protein, you are generally covered. Now there is a condition, genetic condition called PKU, phenylketonuria, right? Or just PKU is fine if you remember it. It's something that you're born with. If you don't have PKU at birth, you're not going to catch it, or you're not going to like develop it later on in life. Okay, it doesn't work like that. So one of the first things they test on newborn babies is PKU. They take a bit of blood from their tiny little feet, and they test for this condition. If a person has PKU, they cannot metabolize phenylalanine. Okay? And you can see here, phenylalanine is one of the essential amino acids. And so the stuff will build up in the brain and interfere with development. Okay? That's not a good thing. Okay? It could lead to quite a bit of problems later on in life. So you want to find out sooner rather than later whether the baby has PKU or not. If they do have PKU, they cannot drink formula. They cannot drink regular formula. They cannot drink breast milk because breast milk also has phenylalanine in it. They have to be put on a special formula that does not have the phenylalanine amino acid in it. Okay, so there you go. It's a little bit of a connection to real life application. Now, I'm not going to ask you to draw anything on the quiz or test. I think I told you that before. But I do want to draw the general structure of an amino acid to show you what it looks like, so it will help you understand a little bit better. Okay? At least I hope it will. So I'm just going to draw it next to this right here. Okay, so the amino acid looks like this. There's a C connected to an H. And this is true for all 20 amino acids. And there's a C connected to H. And it will connect to this thing here, C with a double bond O and OH. And we have a name for this group of molecule. I know I keep on saying this, but you actually will learn this in chemistry eventually, next semester. This thing is called a carboxyl group, which makes it an acid. Give you a moment to catch up on that. And then on the other end, it's connected to N and H. This here is what we call an amino group. So that's why it's called an amino acid, because it has an amino group and an acid group, amino acid. So all 20 amino acids will have this backbone structure. What makes them different from each other is the fourth thing. Remember, carbon can attach to 4 things, right? 1, 2, 3, 4. So this fourth thing here, people just put R, and R can be different things. In fact, it's going to be different for each of the amino acid. We call this the side chain. And it is unique for each amino acid. So there are 20 different types of these amino acids. Each of them have the same core backbone, but the side chain will be different. Let's take a closer look here. This page lists some of the amino acids. I don't know if there are actually 20 here, okay, but there are definitely some examples of it. And you can see, I know it does look a little bit different here on our drawing. It's like OH here, but this one is missing an H. Don't worry about it, okay? For our purpose, they mean the same thing. So, you know, you have the central carbon, you have the carboxyl group and the amino group, and the side chain is different for each. For glycine, the amino acid glycine is an H. For alanine is a CH3. For valine is this, and so on and so forth. So each of the amino acid has its own unique side chain. That gives it a unique property. And we can group amino acids together based on the kind of properties that it has. So sometimes they are non-polar. Sometimes they are polar. Sometimes they are going to have a positive charge, or they will have a negative charge. And you know, grouping them in different categories will help us understand not only their properties, but like where do you expect to find them in a protein, for example. Okay. So how do we connect these amino acids together? Okay. We have 20 different options, and connecting them, it's kind of like building like a necklace with beads. Okay, think of a tray of beads with 20 different colors. So if I ask you to build a necklace, like you will use like the red one and the green one and the blue one, maybe you use another blue one, and so on and so forth, and you make a whole necklace, right? That whole necklace, each bead would be like the amino acid, but the whole thing would be like a protein. Okay, it's a polymer, so we sometimes call it like a polypeptide. It's coming up, okay? You don't have to write down at this moment. Do you understand? Do we have to memorize these? No, please don't memorize these things, okay? For the love of God, do not memorize these, okay? Don't even have to memorize the structure of the amino acid. Although, if I give it to you, you should recognize it, but I'm not gonna ask you to draw it out, okay? Now, what was I talking about? I lost my train of thought. Yeah. So building it, it's like connecting the beads together and having a necklace, right? So the whole necklace is like a protein, and each bead is like an amino acid. Do you, like, if I give you 20 color beads and I ask you to make a necklace, like, do you have to necessarily use all 20 different colors? No, right? Like, do you, can you use repeats? Of course, like if you really like red and green, maybe you will use more of them, right? So in the same way, a protein does not necessarily have to have all 20 amino acids in them, right? It's like a combination of some of these amino acids, and it's okay to use repeats. Some proteins are, you know, 10, 20 amino acid long. Others are hundreds of amino acids long. Okay, it really depends on what protein you're talking about. Okay, so how do we connect these amino acids together? Well, let's try to draw that out in the note here. And I think this part is best if you don't draw along. Okay, you can draw along later if you watch the recording, but I just want you to see it so you understand it. Okay, so again, don't draw it with me. Just watch what I'm doing and try to understand it. Okay. So here I am drawing an amino acid. And the side chain, it could be any one of those things that I showed you, right? So I'm just going to call it R1, side chain number 1. So this here is an amino acid. Do not draw this, just watch. Now I am going to be lazy, and I'm going to copy this guy and put it right here. So here is going to be another amino acid, and it would have a different side chain, right? R number 2. So this is amino acid number 1, amino acid number 2. How do we generally connect things together in this unit? What do we need to take out in order to form a bond and join these guys together?
Water.
Water is correct. Okay, water is correct. So what we will do is we will take an OH and an H, And that's going to come out as H2O. Right? And more importantly, after you remove the OH and the H, you can connect these two things together. And this new connection, this new bond that you just formed is called a peptide bond. Peptide bond. Okay? And you can understand how you can go on forever. Right? Let me just put another guy here. Like if you wanted to connect to amino acid number 3, no problem. You take away the OH and you take away the H, you remove another water molecule and you form another peptide bond, and so on and so forth. Does that make sense? Any questions at all?
So the peptide bond is a covalent bond or
Yeah,
ion?
absolutely. It's a covalent, it's a sharing of electrons. All the bonds in macromolecules that we are learning, all of them are covalent bonds. All of them are sharing of electron. Okay, why don't we take a quick break? We will come back at 1:37, and we'll do the rest of them. Okay, see you in a bit. Hello, hello, it's me again. Let's keep going. So, that's how you connect the amino acids together, and that's how you eventually build a protein. And like I said, some proteins are just a couple amino acids long, others are, much longer than that. One of the important things you have to understand when we discuss protein is its structure. Right? There are 4 levels of structure associated with the proteins: primary, secondary, tertiary, and sometimes you might have a quaternary structure, a 4th level, but not for all proteins, just sometimes. Let's take a closer look at each of these levels. The primary structure refers to the unique amino acid sequence in the polypeptide. Again, polypeptide is just another word for protein. Just like for the, carbohydrates, we say polysaccharide, right? It's just to remind you that it's a polymer. That's why we have this alternative name for the macromolecules. Now, if you look at the amino acid sequence of each protein, it is going to be unique. It is going to be different. I mean, if it's the same protein, then it's going to be same. Like all insulin is going to have the same sequence of amino acid, but the sequence for insulins are going to be very different from, say, like glucagon, right? It's different.
I have
Hormones.
a question.
What's that again?
I have a question.
Sure, please.
So what is peptide finally?
Oh, peptide. Because like when you connect them together, you have a peptide bond, right? So that's why it's polypeptide. It's just a bunch of amino acid linked together, forming a polymer connected by peptide bond. Peptide, basically protein. Okay. So, like our analogy, if you have the beads here, and I know there are more than 20 different colors, but use your imagination. You build the necklace, and you read the color from one end to the other end of your bead. That's basically the primary structure. Now, what determines the amino acid sequence in the protein? Actually, that is encoded in your DNA. That is encoded within your DNA. Your DNA, amongst other things, is going to give instruction on how to build these proteins. It's going to tell your body what amino acid to link together. So after you link all the amino acid together, you get a strand of polypeptide, like this thing right here. Okay, if you can't see me on the video, please turn on the video so you can see me, because I'm showing a prop here. Now I know I don't have the beads on it, because if I put the beads on, it's going to be like too cumbersome. I cannot manipulate it. Okay, but you get a strand like this with the amino acids on it, and depending on the kind of amino acid you have in the sequence, some parts of it is actually going to form these helices. Okay, so it's gonna kind of like this. That's a helix. And then, another part of it might also form helix. So the secondary structure is basically describing whether the protein has helix in it, okay, and like how much of it is going to be helix form. Or instead of forming the helix, sometimes they would form these zigzag. Okay, so just up and down like that, zigzag. The up and down, they are called beta pleated sheets. And then the helices, they're called alpha helices. These are Greek alphabets, okay? Alpha and beta. And on diagrams like these are called ribbon diagrams. It's a standard way of representing proteins. The alpha helices are represented by the coils, and then the arrows, they represent the beta pleated sheets. Okay, so these things are generally stabilized by hydrogen bonds of the backbone. And a protein might have all helices, or it might have all beta pleated sheets, or it can have a combination of both. Okay, so helices at one part, beta sheets at another part. Okay, so secondary structure is basically describing, how much of these things you have in the protein. Just on a side note, like why do we care about these alpha beta stuff? Well, mad cow disease. Actually, in my other classes, a lot of people have not heard of mad cow disease. Anybody know what this is? Mad cow disease. Like, how do you catch it? Anybody know?
Yeah, it's in the cow.
Yeah, as the name implies, it's from the cows, right? And you catch it by eating contaminated cow meat. Okay, so in our body, you and I, we have this protein called prion proteins. Prion proteins. Okay, it looks like this, and you can tell from the ribbon diagram there are like alpha helices in it, right? Alpha helix. And what happens is in cows with mad cow disease, their prion proteins, it's the same sequence as the normal ones that we have, but the folding is different. So instead of just alpha helix, you can see it has these parallel sheets, right? So there are beta sheets in it instead. And so this is what happens. If you consume contaminated beef, you will have the misfolded prions in your system. And then these misfolded proteins will find the normally folded protein, will find the normal proteins, and then it will cause it to misfold. Okay, so now you have two misfolded proteins. They in turn would go find more normal protein and cause them to misfold. And that quickly spread and cause your prion proteins to switch from the alpha form to the beta form. And this happens in the brain, in the central nervous system. And when enough of the misfolded proteins are there, they will clump together and cause the neurons to die. You are not supposed to have holes in the brain. And this is the brain tissue of someone who died from mad cow disease. You can see like there's so many holes. All these are holes, okay? And the holes is basically what kills the person. It's the death of the neurons, right? So if you look up mad cow disease, the medical name for it is spongiform encephalitis. Encephalitis is inflammation of the brain, and spongiform, as you can probably guess, means that the brain looks like a sponge, a lot of holes in it. Okay, so by understanding the configuration of the protein, right, people can try to understand why it misfold and maybe come up with an effective treatment for people who are infected. By the way, like you cannot cook your meat and think it's okay, right? Because this is not a virus, this is not a bacteria. Cooking is not gonna kill it, right? It's a protein. So even if you cook the food, it's still gonna be contaminated. All right, tertiary structure. Now, if you take the protein, right, and you fold it up into like a ball. I mean, it doesn't have to be a ball. It could be a cylinder. It could be other shape, but fold it up into a three-dimensional structure. The 3D shape of the protein is called the tertiary structure. These are stabilized by interactions between the side chains. Remember, each of the amino acid has a unique side chains, and different side chains, when they come close to each other, can interact in different ways. Some of them can form a bridge, some of them can form an ionic bond, and so on and so forth. You do not have to memorize these interactions, but know that they are what supports the 3D structure. Right? The primary structure is held together by covalent bond. The secondary structure are held together by hydrogen bonds, and the tertiary structure are held together by various various interactions between the side chains. There is a question about this in the study guide. There's a table that you can fill in. Please look at the answer key for that. It will summarize all I've said for you. Now, most protein are gonna stop here. It's complete, it's fully functional. This guy can go on and do whatever it's supposed to do. However, in some cases, in order for the protein to be functional, like on its own, this green one cannot do anything. On its own, the pink one here can't do anything. They actually have to partner up and come together. They join together to form what we call a protein complex. And so like this whole thing here, that is a protein complex. And this particular one on the PowerPoint slide has 4 different peptides in it, 4 different polypeptides. Okay, so 4 proteins come together to form a bigger protein. What components you need to put together to form this bigger protein? That's basically the quaternary structure. Okay. Any questions about the different levels of the proteins? Okay, if you think of anything, feel free to email me. I haven't been receiving too many emails from you guys regarding questions. Please note that.
The primary structure is functional?
No, the primary structure is just the sequence. It's not fully functional until you fold it up into the 3D shape. Okay, so the primary level is going to determine how much helix or sheets you have, which then in turn determines the overall 3D shape. So you got to build it up one layer at a time. By the way, I don't know if you guys seen the announcement, but your tuition includes complimentary tutoring services, right, from the library. So you can book an appointment with our co-op tutor. His name is Kian Leung, and he can help you with biology, chemistry. Okay, I think there's a separate tutor for math, if I'm not mistaken. But you know, if you need extra help with the bio stuff, he's the guy to go to. These tutors tend to get booked up pretty quickly. I mean, like you look at the time, you're like, oh, 10 to 5, that's a lot of time. But trust me, like people book them up, especially right before a quiz. So if you anticipate you need help, I suggest you go and book an appointment sooner rather than later. Of course, I'm available for help as well, so make sure you take advantage of that also. All right. Moving on, the importance of protein structure. Each level of protein structure contributes to the overall shape, as I told you. You got to build it up one level at a time. Okay, if you made a mistake in the first level, if you made a mistake with the primary level, you might end up having a different protein in the end. Okay, sometimes it's okay, but sometimes it's not. Usually it's not okay. The overall shape of the protein has a name. Right? We call it conformation. Right? In biology, people like to use big names for simple things. It's not my fault. It's just the way it is, okay? So the conformation, the shape of the protein, it's very important for its function. Right? We usually say there's a key and lock relationship between the shape and what it does. Because if you change the shape of the key, you cannot open the lock. If you change the shape of the protein, it will no longer work as expected. So there are different factors that would cause the protein to change its shape, to unfold. When that happens, we say the process is called denaturation, and we say the protein has been denatured. Okay, so denaturation basically means unfold a protein. So you can unfold a protein by changing the temperature. Too cold or too hot can both cause the protein to misfold. Okay, I'll give you two examples. Egg has proteins in it. Okay, it's in the egg white, and when you cook the egg, you are denaturing the proteins in the egg white. It goes from translucent to white. You can't go back. It's a one-way process. Jello is basically proteins, okay? And like when you make Jello, right? Like you have a pouch of these protein powders, and then you add the hot water to it. It dissolves, and you put it in the fridge. And putting it in the fridge is going to cause it to cool down, and then the protein, right, becomes like a solid state. Right? You can't really melt Jello. Okay, it doesn't really go back to the liquid form. So again, the transformation, it's like a one-way process. Cooking meats is the same thing, like changes color from red to brown. That is a denaturation of the protein. You can also denature proteins by changing the pH, salt concentration, and type of solution. That's why it's like dangerous to have prolonged high fever, because the high temperature can actually cause some of the proteins in the body to break down and therefore not function properly. Here is an example of a condition called sickle cell anemia. This is something you're born with. If you don't have sickle cell anemia at birth, you're not going to develop it later in life. You can't catch it. You cannot catch it, okay? So in the body, in the red blood cells, that's what they look like. Like these are actually red blood cells, okay? Red blood cells. They look pink because, you know, we dyed them a certain color. The normal ones are the circle ones here. Okay, so that's a normal looking one. That's a normal looking one. These ones are not normal. Okay, these are sickle cell shaped. They're banana shaped. Okay, so what caused that to happen? Well, inside the red blood cell, there is a protein called hemoglobin. Hemoglobin. Hemoglobin is inside red cells, and it helps transport oxygen and carbon dioxide. Okay. Hemoglobin is made up of 4 separate polypeptides. Okay, like 4 of these things all come together, forming the hemoglobin. And one of the polypeptide has 146 amino acids in it. Okay, so the first 7 goes like this, right? There's valine, histidine, leucine, so on and so forth. You don't have to memorize the names, okay? But at number 6, the normal hemoglobin is going to have this amino acid called GLU, okay, glutamic acid. But for people who have sickle cell anemia, the instruction to make the protein, the DNA is the instruction, right? So there is a mutation in DNA which caused the instruction to be incorrect. And when the body tries to make the hemoglobin, number 6, instead of putting the glutamic acid, they put valine instead. And that causes the hemoglobin to have a different shape, a different conformation. And that in turn causes your red cell to have a different shape. These sickle cell shaped red cells cannot transport oxygen efficiently. They get stuck in capillaries, and it will cause the person to have shortness of breath all the time. They will feel tired, fatigue, and sometimes when they get stuck in the capillary, they could cause quite a bit of pain as well. Okay, so this is one of the conditions where they're trying to fix with gene therapy. Right? By giving them the correct DNA, maybe the person can now start to make normal hemoglobin again. So that's something we'll look at later on in the course. Lecture 9, okay, for gene therapy. Okay, what other functions of proteins do we have in the body? They form important structures like muscle, the 2 key muscle proteins that we have. Nails are made up of proteins, hair, Right? We don't have horns, but like horns of other organisms are made up of proteins. In fact, the horns are made up of the same kind of stuff that makes up our fingernails. Okay, spider web is actually proteins. Proteins also made up hormones. Right? Earlier we talked about steroids being hormones, but not all hormones are steroids. Some are protein. Right? Insulin is an example of that. They are a good source of energy, although we do prefer carbohydrates, but protein is also usable as a form of energy. That's why we eat meats after all, right? Okay, or like that's why you need to have proteins in your plant-based diet, right? Because we do need them as energy source and to use them to build all these things in the body. And it also can be an enzyme. Right? More on enzymes later. On a side note, animals like rhino and this thing called pangolin, they roll up in a little ball. Okay? You might have seen that before. They are on the brink of being extinct because poachers hunt them down. And one of the reasons why they hunt them down is to take the horns or take the scale and they sell it on black markets because there are a lot of people who believe there are medicinal values to some of these things. Okay, rhino horn, for example, are sometimes grind down into powders, and people believe that it's an aphrodisiac. Basically, it means like it increases their sex drive. Or some people believe that the scale of the pangolin is useful for treating things like asthma. Okay, which is not true. Based on what I told you, the rhino horn and the scale has the same kind of protein that makes up our what? Do you remember? Information
Nail.
overload, eh? What's that? Sorry, Elvis, go ahead.
Nail.
Nails, that's right.
Yes.
Okay. Now, nails, right? Look, if you bite your nails, like does it change your sex drive, right? Like probably not. And if it does, like I don't want to know about it, right? Does the nails treat you for your asthma? No, right? Okay. So like people need to educate themselves on these things. Okay. Don't just believe things that they find online or people tell them about, right? In fact, the media coverage was what's driving some of these misinformation. Okay. They are just the same kind of protein as what you make in the body. Right? And they have no medicinal value whatsoever. So like, the rhinos and these things are going extinct for no good reason. Um, anyways, the last thing on the list of protein function is enzyme, right? And let me just spend a little bit of time talking about this. Enzymes are proteins that catalyze. That's another big word for speeding up. Okay? They speed up chemical reactions in living things. It turns out chemical reactions don't really happen for free. Okay? You need to invest a little bit of energy for a reaction to happen. Okay? Think of like skiing down a hill or going to tobogganing, all right? You actually need to go up the hill first before you can have fun coming down, right? And that's kind of what happens with reactions in the body. Okay? In a chemical reaction, you start with the reactants, which are like the ingredients, and you get the products, the end result stuff. But you have to go up an energy hill first before the reaction can happen. So in an uncatalyzed reaction, in a reaction that does not use enzyme, the energy required to start the reaction is actually quite high. Okay? This energy, EA, is called activation energy. Okay, and it's quite high. It's not possible for our body to supply that kind of energy for all the reaction that takes place in the body. So instead, we rely on enzymes. Okay? Enzymes allow the same reaction to take place, but with lower activation energy. Okay, it helps them a little bit. So like It's kind of like the pre-health program, right? Like if you try to go into a nursing program on your own, you need at least like 80% grade or 85% grade. It's very difficult. It's not impossible, but it's difficult. But if you go through us, right, you only need to meet a requirement of like 70%. Okay, so it allows the same outcome, but with less energy. All right. So here is an example of an enzyme and what it looks like. There's a lot of like big words here, but basically it goes like this. Here is an enzyme. Okay, and you know what? I actually might redraw this for you. It might be a little bit easier than like a 3D picture there. Okay, so like this is an enzyme, and the enzyme has a pocket. Okay, so this pocket is called an active site. Okay, you don't have to redraw it. Okay, you can just watch and understand. It's the same information on the slide anyways. So the pockets, the active sites, that's where you will fit the ingredients into it. Okay, so like I have a square that would go into this slot. I have a triangle that goes into this slot. So the pocket has to be the appropriate shape for the substrates. Right? Substrate is just another word for the ingredients. The reactants, okay? So if it's like a different thing, it's not gonna fit in, okay? It's not gonna fit. It has to be the right shape. Okay. And this here is our enzyme. And that's what I mean when I say that the enzyme, which is a protein, has to have the right shape. If you change the shape, it will no longer work. Because if you heat up the enzyme too much and the pocket changes shape to like a circle, then it won't work anymore. It's not going to fit the ingredients. Right? So what happens is these things goes in, and then afterwards, the enzyme is going to do the reaction. It's going to connect them together, and you have your product. Okay? So that's essentially what it does. So for example, if here we have a glucose, and then we have a fructose here, it goes into the enzyme, it goes into the pockets, the active site of the enzyme. The enzyme will do a reaction and it spits out the product. You should know glucose and fructose will give you sucrose. Okay. Similarly, if you want to break down sucrose, you need another enzyme for it, and then it will spit out glucose and fructose. Different reactions in the body require different enzymes. Almost all the reactions that happens, it's going to need its own enzyme. All these enzymes are proteins, and the instruction to make them are in the DNA. As you will see, if you made a mistake in the DNA, some of these enzymes might not work properly. And a lot of the diseases that we see in humans is at the root of the problem is because we are making the wrong enzymes. Okay, or we're not making the right one. Sources of protein. Meat, I told you, is excellent for vegans and vegetarians. Beans, nuts, they are all rich in proteins. Okay, soy product, right? Like tofu, soy milk, almond milk. And that's it. Okay, again, we don't have time to go through these chunks of track questions. I will post the answers for you later on. Okay, we have about 15 minutes. I think I probably will need 20. And then we are going to finish the section on nucleic acid. Okay. Anybody know what these things are? These like little gummy worms that you're looking at, colorful ones?
Chromosome.
They are chromosome. That's right. Chromosome. Zome is body. Chromo means color. Okay. These are bodies that will soak up color. That's why it's called chromosomes. Okay. How many chromosomes do you have? No one has been able to answer me so far in my other two classes. Let's see if you guys know. How many chromosomes do you have in your body per cell? Okay, we have 34. We have 23. Anyone else?
46.
46. We hit the jackpot right here. Okay, I think Jericho, you mean 23. 23 pairs, right? 23
yes,
pairs. Yeah.
23 pairs.
Yeah, that's right. 23 pairs and 46 chromosomes in total. 23 from mom and 23 from dad. The egg carries 23 chromosomes. The sperm carries 23 chromosomes. When they come together, we get 46 chromosomes. Okay? We will learn a lot more about DNA later on in the course, but for now, let's just go through some of the basics. Okay, if you still have the class note, please pull it out. We're going to go back to this table that we haven't not finished. So we're at nucleic acid now, the last thing in the macromolecule lesson. And the element that makes it up is, again, the standard CHO and NP, nitrogen phosphorus. Okay, CHO, NP. The monomer for nucleic acid, right? The building block for a nucleic acid is something called nucleotide. Nucleotides. Okay. There are only 4 types of nucleotides. Sorry, 5 types. Okay. A, C, G, T, U. Yeah, thank you. As time goes on, you will remember what they stand for because you'll be talking about it like all the time. For now, it's okay. You can just memorize the like the alphabets, okay? A, C, G, T, or U. Okay. So what is a nucleotide? Right, so this is the general structure of an amino acid. The general structure of a nucleotide goes like this. Okay, it has 3 parts to it. There is going to be a phosphate. It's the same phosphate as the one in phospholipid, okay? PO4 -3. There is going to be a sugar, which is represented by the pentagon here. And also, we are going to have a base here. Nitrogen base. Okay, sometimes they call it nitrogenous base. It doesn't matter. Okay, so the whole thing is a nucleotides. And there are 5 different kinds of nucleotide depending on what kind of base it contains. The base here can be a C, a G, a T, or a U. Okay. The sugar and the phosphate are the same for all the nucleotides. It's just the ACTU that makes them different. The base that makes it different. Right. Just like the amino acid, same core structure, the side chain is what distinguishes them. Okay, so there are 2 types of nucleic acid. This is the one that most people are familiar with. It's called DNA. The D stands for deoxy, and then ribonucleic acid. Whereas RNA is just ribonucleic acid. So as the name implies, DNA, it's deoxy. So is missing an oxygen. Okay, missing an oxygen atom. I'll tell you where it is in a second. So here we have 2 nucleotides. We have the phosphate, we have the sugar, and we have the nitrogen base. Okay, so want you to pay attention to the sugar. On carbon number 2, we have an OH, and on carbon number 2 here, we have an H. So this one is deoxyribose, is missing an oxygen, right? So that would be a nucleotide for DNA. Whereas this one is a nucleotide for RNA. So one of the differences between DNA and RNA is the kind of sugar. Like, is it a ribose sugar or is it a deoxyribose sugar? The base is also different. For DNA, it is AGCT, whereas for RNA, it is AGC or U. So both of them have AGC, but DNA has T only, and RNA has U only. Questions? Let's take a closer look at the nitrogenous base. We can separate the base into two groups, purines and pyrimidines. Okay, purine, pyrimidine. The purines, they have two rings in their structure. I remember, like when I was studying this in school, I always got confused. So like purine has two syllables to it. That's why it's two rings, okay? And pyrimidine has one ring. That's how I remembered it. And you should know which ones are the purines. So A and G are purines. A stands for adenine and G stands for guanine. Okay, so to remember it, you can remember this phrase, pure as gold. Pure as gold. So purines are A and G. Adenine and guanine. Everything else is pyrimidine. A and G. Purine. Two rings. Okay? So the nitrogenous base is part of the nucleotide, right? The nucleotide is the whole thing. The phosphate, the sugar, and then we got the base. The base can be one of these things. A, G, C, T, or U. Remember, T is only in DNA, and U, uracil, it's in RNA only. The DNA molecule is like a ladder. Okay, think of a ladder that you can climb on, but you twist it up. Okay, you twist it up so it becomes like a spiral. Right. So the rocks of the ladder that you climb on, that is basically the nitrogenous base. Whereas the side of the ladder, that's the sugar phosphate backbone. If you look at the diagram here, the blue and yellow pattern, that's the backbone. That's the side of the ladder. Okay? Yellow is the phosphate, blue is the sugar. So it's just sugar, phosphate, sugar, phosphate, sugar, phosphate all the way down. Now I want you to pay attention to the base. Okay, help me out here, guys. A, is that a purine or is that a pyrimidine? That's right, it is a purine. Okay, A is a purine, pure as gold. Remember that, okay? So it has two rings in it. T is not part of the pure as gold phrase, so it's a pyrimidine, and it only has one ring. So across here, it is going to be 3 rings wide. And what about cytosine, C? Is C 1 ring or 2 rings?
1.
1 ring is correct, okay? And G is guanine, 2 rings. So again, across we have 3 rings. And if you check all of them, it turns out it is always going to be 3 rings wide across, okay? And that's important when you're building a ladder. You don't want it to be like 3 rings wide sometimes it's 4, sometimes it's 2. If that's the case, your ladder is going to be like bulging in some places, and then it's going to be like super narrow in other places, and then it's like all over the place, and it's going to make it unstable, right? So we don't want that in DNA. We want the double helix, we want the DNA to be consistent width along the whole length. And that's why it follows the base pairing rule. You will see that A always pairs with T and G always pairs with C. In this picture, you can see 2 halves of the DNA. Okay, we have the phosphate and the sugar, back and forth, okay? And T and A are pairing together, okay? These little dotted lines, those are what's holding them together. Okay? Hydrogen bonds. Okay? 2 hydrogen bonds holding the A's and the T's together. 3 hydrogen bonds holding the G and the C together. Okay, 2 hydrogen bonds, 3 hydrogen bonds. Okay, and we have the sugar and phosphate backbone. Now, knowing the base pairing rule is going to allow us to predict what's on the other strand. This predictability, we have a name for it. It's called complementary. Complementary. Over here, if you study the diagram closely, you will see that one strand is actually upside down relative to the other side. And that's the second property of DNA. It's called anti-parallel. Right? We'll talk more about this later on. For now, just think of like the highway. Right? The highway has like a northbound and a southbound, right? Like think of DVP or something. Right? The 2 lanes are parallel to each other, but the traffic runs in opposite direction, right? That's what anti-parallel means. Parallel, same distance between each other, but it's opposite in direction. So DNAs are complementary. You can predict the missing half if you know half, and then it's anti-parallel. So here we have one strand of DNA. Using the base pairing rule, we should be able to figure out the other side. Okay. So what does A pair with again?
T.
A pairs with T. Okay, you can remember it spells at.
At.
Yeah, and C pairs with G.
G.
Okay, so over here, if you see A on one side, the other side must be a T. If you see G, the other side must be a C. And using the same rule, that A goes with T all the time, and T goes with A, and C goes with G, we can predict the missing half of the DNA. This predictability is one of the properties of DNA. We say DNA strands are complementary. Complementary. Are we okay? Everybody still with me?
Yes.
Thanks, Elvis. Okay. Hopefully the rest of you are also with me. Now here is another question. Okay, thank you guys. Okay, suppose the cell has 40% adenine. Find the percentage of the other stuff. Now listen, if you have, I should add, okay, 40% adenine in DNA, okay? If you have 40% adenine in the DNA, you will also have 40% of what else?
T.
Yes, because A goes with T all the time, right?
Yeah.
So 40% A must be 40% T. If you add these things together, you will get 80%.
Yeah.
Now you cannot go beyond 100%, right? So
Correct.
100 minus 80 gives you 20%.
20.
Yeah.
So 10-10.
Exactly. So the 20% is split evenly between the C's and the G's. 20 divided by 2 gives you 10%, 10%. Okay. So knowing the percentage of one of the 4 bases will allow you to quickly calculate all of them. Okay, now just a quick one for those of you who need a second example. 30% C. If you have 30% C, you will have 30% G. Together, they add up to 60%. 100 minus 60% gives you 40%. And the 40% will be split evenly between the A's and the T's, which means it's 20% and 20%. Okay? That's all.
Do we have an example of anti-parallelism?
Oh, yeah. So for that, it's not important at this point, okay? All you need to know at this point is that what it means to be anti-parallel, okay? Parallel, but opposite direction. When we get to lecture 6, that's going to be more important, and then we'll explain it when we get there, okay?
Okay.
Finally, last slide, RNA. Okay. RNA differs from DNA in that it is not a double helix. It doesn't have two sides of a letter. It's only one side, single-stranded. The backbone is also sugar and phosphate, but it's a different kind of sugar. The DNA one is missing an oxygen, so it's called a deoxyribose. But this one has the oxygen. It's just a ribose, okay, pentagon. The nitrogen base can be A, C, G, or uracil. There is no thymine in it. There are different types of RNA. They are all listed here. We will learn about the function of different types of RNA when we get to lecture 7. For now, just be aware that there are different kinds. You don't have to know what they do yet. And that is it. Okay. Again, there are more multiple choice questions for you to try. I probably will have time to take it up at the beginning of next class, okay? So just come on time. I mean, definitely try them out first, and then come on time next time on Wednesday, and then we'll take it up first thing before we start on lecture number 3. Okay, so please do print the notes for that and start studying for the quiz, which is going to be next Monday, and I'll post the information about the quiz shortly today. All right, thank you for your attention. Have a good one, guys. Bye for now.
Thank you.